so a couple of quick notes. Progress reports, I've seen almost all of them in the Dropbox or sent to my email already, so thanks for sending those in. My goal, 24 hours, they're back in your hands. That's my goal, is to get these back as soon as possible so that you've got my feedback to be working on the poster. Tomorrow/Wednesday, I'd like to see a draft of the poster. It's a 4-point assignment. The points are there just to kind of keep you guys motivated to do it. I know that not everybody is complete with their work, and that I understand going into this. But I'm looking for, really what I'm looking for more than anything else is that you have a complete layout and you know where you want everything to go. Okay. The final draft of those, I would like to make the deadline Friday night, if possible. What I'm going to do is I'm going to get my feedback to you on those posters again within 24 hours. So the sooner you get it to me, the sooner I can get it back to you, so that you have time to make changes. And then when it comes to submitting the final one, what I'd like you to do is 2 things. 1, of course, submit a draft to me. 2, I've put up the instructions on with the poster information itself on how to submit this to TAC to get it printed. Make sure you do that as well, otherwise it doesn't get printed in time. So, and I honestly don't know how these are being paid for, so because I think in the past biology has taken care of it. If not, we can pay for it off the grant. So no cost is going to you guys on this. So what I'd like you to do is, as you're putting in the information for the poster and everything, you're going to probably want to make sure that they know, of course, the size of it so it doesn't get scaled incorrectly. But the other thing you'll want to do is just make sure that you note any questions regarding payment. Just refer them to me, and I will take care of it. Okay, so just refer them to me if there's any questions about payment for it. And I'll either get them the grant number or Bio will pay for it, one or the other. Okay, so that's about it for right now. Lit Review Wednesday. So it's kind of a hectic week, but finally, once after this week is done, I think things will start to, at least in here, quiet down a little bit. The plan is that Friday we start talking about HIV. We'll talk about that Friday, Monday, Wednesday. Next week, Friday, we have off for the presentations. And then that following Monday and Wednesday, we'll finish up with HIV. If we're going to do vaccines, I've decided to just toss that aside for now. I'd rather spend 3 days on it than 1 day, and I'd rather spend no days than 1 day. So we'll just skip it. So we will have one more assignment coming up down the road, either another critical thinking or a lit review. And let's see, the 4th is our last in-class exam, and around that time I'll be giving home the last take-home as well. Okay, so that's kind of where we're at. We've only got 4 weeks left. So, what's wrong with that, Sabrina? Well, yeah, I hear you. Alrighty. Alright, so what I wanted to do for today's topic is to cover, with Ebola, some of those other things that it does to actually suppress the immune response. I preface this with a lot of our understanding of how it suppresses the immune response is based largely on studies done in petri dishes rather than in organisms. Again, if we're working with it in organisms, we have to adapt the strain to the host. And we can do that fairly easily because it is an RNA virus, and because RNA viruses do have a higher rate of mutation than do the DNA viruses. So we're going to focus our discussion of this on pretty much one single protein, VP35, and its ability to block interferon signaling. We also talked, last time we discussed this, which was about a week ago, we talked about soluble GP and its role in blocking antibody. So just to kind of now go over some quick background on interferon. This is probably one of the most, this is a very well-studied innate immune signaling pathway. Interferon itself is named in part because it interferes, and it interferes with viral replication or viral life cycle. The molecule itself, interferon, there's type 1, which consists of alpha and beta. Type 2 is gamma. We're going to focus just on interferon beta here. And the thing that I want you to know about it is a signaling molecule. It is secreted by macrophages and other cells when infected. And when those cells identify a foreign substance either on the surface of the cell or the inside of the cell, and when it is secreted, it's going to bind to interferon receptors, or IFNRs. And these are going to be located on the surface of pretty much every single nucleated cell in the human body. When this binding occurs, it triggers a signal cascade that's going to lead to events like repression of PKR, activation of PKR, upregulation of P53, upregulation of RNA cell, which will cut double-stranded RNA, unbound double-stranded RNA, and then MHC1 within T cells, and MHC1 on any nucleated cell, and that's going to be used by T cells to recognize foreign substance inside of those cells. So the goal here is really to activate not only a cell's innate immunity, but also to activate an overlap with the adaptive immune response as well. So VP35 blocks a couple of steps along this process, and that's what we're going to talk about today. One that we'll talk about is VP35 will actually block identification of double-stranded RNA by a protein known as RIG-I. RIG stands for retinoic inducible gene, or retinoic X inducible gene. So the name's not all that informative as to what it does, but it recognizes double-stranded RNA. We'll talk about this a little bit more in a few minutes. VP35 also blocks this phosphorylation step of these interferon factors, or interferon, these are transcription factors that are going to bind to interferon promoters and activate transcription of interferon beta.
Can you say that again?
Yeah, so VP35 is going to block the phosphorylation of these IRFs, which are transcription factors that activate the transcription of interferons and other innate immune genes as well. Now, the way that VP35 does this is not only is in a couple of ways. One, VP35, as well as nucleoprotein, they bind to the viral genome. And in binding to the viral genome, there's going to be a point at which that viral genome is double-stranded during the process of replication or transcription. So binding up the double-stranded RNA is going to prevent its detection by proteins like RIG-I, there's another one known as MDA5, and PKR, that all are induced by the recognition of that double-stranded RNA. Okay? So nucleoprotein and VP35 basically kind of serve as like cloaks or masks to prevent the identification of that double-stranded RNA. So that's mechanism one. The other is that VP35 directly interacts with cellular proteins to inhibit their function. And that's what we're going to focus on. Okay. We'll see a third example a little bit later on, but the other thing that VP35 does is it inhibits PKR activity. So those are three things that it does. Inhibits PKR, inhibits RIG-I, inhibits ERF3 and 7. So the evidence for this started accumulating around the early 2000s, late 1990s, when groups such as Basler's group, and he's kind of one of the experts on Ebola virus and understanding these immune responses. His group was looking at the ability of VP35 to inhibit interferon signaling, or to inhibit interferon activation. So this is an experiment in which his group took just 293 cells, transfected them with a plasmid expressing the interferon beta promoter upstream of chloramphenicol acetyltransferase. So this is analogous to like the luciferase assays that we've talked about in the past. These are reporter assays. Chloramphenicol acetyltransferase is present. That must mean this promoter is active. Okay. Put that, and then they put that in with 3 different vectors, one containing nothing, one containing a protein known as NS1. This comes from influenza. And then VP35 from Ebola virus. 24 hours later, transfect these cells additionally with poly-IC, which we've talked about mimics double-stranded RNA. So they're trying to induce a double-stranded RNA response. They're measuring, though, interferon beta promoter activity in the presence of nothing else, NS1 or VP35. Everybody following so far? So this is just reported as a fold induction, or how much chloramphenicol acetyltransferase is being expressed. Alone, when poly-IC is added, we get about a 14-fold increase in the amount of chloramphenicol acetyltransferase activity. So this promoter is active in the presence of poly-IC. What does NS1 do to it? Activate, repress, inhibit. Just compare these two. Does NS1 inhibit the response, activate the response? It inhibits the response. Okay. So what this says is that influenza's NS1 protein inhibits interferon signaling. What about VP35? Again, compared to the control, or to this one, this sample here. Still an inhibition, okay? So at least what this evidence said was that VP35 had the ability to block interferon beta expression. It wasn't known how. And so that's where then the research has been going over the past few years, has been to understand, well, actually, how does it block this interferon beta expression? Okay. So let's take a look at a couple of examples here. First, we're going to look at RIG-I. So RIG-I is a protein that consists, you can think of it as containing 2 different domains. One is a helicase domain, and the helicase domain is what binds to double-stranded RNA. Okay. The other domain are these CARD domains, and CARD stands for caspase regulatory domain. The terminology is not all that important, but I'm providing it in case you want to know what it means. You can think of the CARD domains as being the active domains of the protein, and the helicase domain being a regulatory domain. So what do I mean by that? Well, normally RIG-I is found in a model like this, where the CARD domains and the helicase domain are close together, and the helicase domain basically occludes or prevents the recognition of these CARD domains. So think of this in terms of analogy to cyclin-dependent kinases real quick. CDKs are always, or cyclin-dependent kinases are always expressed. When do they become active? What has to bind to them to become active? Cyclin have to bind. And we said that the cyclin changes the shape to open up the active site, right? There's some analogy here. When double-stranded RNA from a virus, for example, binds to this helicase domain, changes the shape of the protein, unfolding this protein or allowing these card domains to now be accessible. So always express, but goes from an off to an on state when double-stranded RNA is binding to that helicase domain. In the next step of the process, these card domains bind to another protein known as MABs. MABs, I don't really know much about it other than it's bound to the mitochondria, but they interact through these common card domains, and somehow then this allows for MABs to serve as a scaffold protein. So it starts to accumulate on it other proteins that ultimately, and this is question mark here, we actually know the answer, but we're just going to leave it as a question mark right now, as a black box of what's going on, because that's not the important part. MABs will ultimately phosphorylate this complex of proteins, including IKK beta and TBK1, or TBK1 and IKK epsilon. So an IKK and TBK. This protein will become phosphorylated. It's a kinase, it phosphorylates ERF3, and that then can bind and activate the interferon beta promoter. If you're trying to keep track of all this, what are the important things to take away? Okay? It's not all of these middle steps. Let's focus mostly on the beginning and the end here. What is the signal that activates this promoter? So go back all the way to the beginning. What's the signal? Double-stranded RNA binding to RIG-I. Good. That's the most important part right now. Okay? So keep that correlation in mind. The other thing that's been found is actually, let me see if we've got this on the next page. Actually, we'll save that for a little bit later. Okay. So I'm going to skip us doing this experiment here. But basically, I'm going to show you some results real quick of an experiment that was done that gave support that RIG-I actually binds to VP35. I added this yesterday. I did not update the slides to include this. So if you'd like it, I'll send it to you. But here's an experiment very similar to the last one we saw. They're measuring interferon beta promoter activity again. And here we're just looking at fold induction or activation of that promoter. In this experiment, they infected with Sendai virus, which is a RNA virus. So it's going to produce double-stranded RNA at some point. And what we find is that normally we get an induction of that interferon beta promoter about 750-fold. Okay? If we then also put in a vector that expresses RIG-I, we can enhance that up to almost 3,700-fold. And now look what happens. Now they start to add increasing concentrations of VP35. What happens to interferon beta signaling in the presence of RIG-I when VP35 is added? It decreases, right? It decreases quite dramatically over this period of time. This is the data that supports this model that VP35 is inhibiting RIG-I. What we don't know from this is how does that inhibition occur? Is it through direct binding? Is it through competition? What we do know is it actually doesn't require VP35 to bind to double-stranded RNA. And that's where this data comes in. So they created two different mutants of VP35, one that contained an arginine to an alanine mutation, another one lysine to an alanine mutation, and we still see repression of this signaling with both of those mutants that are unable to bind to double-stranded RNA. So it seems that at least this activity of VP35 isn't dependent on its binding to the viral genome, but it's dependent on its interaction directly with Reg I. I'm just going to stop there real quick and see if there are any questions. Amelia.
That IRF3 you said binding to the promoter for the interferon? Okay.
Yes. Yeah. Okay. All right, so the second mechanism we're going to look at is PKR in VP35. We're actually going to skip the IRF3 and IRF7 for now. Probably won't come back to it, but that's okay. All right. So let's take a look at some data for PKR. But before we do this, let's just review PKR. What does this protein do? We've seen it a couple of times now. Maybe we can pick up some contextual clues. EIFs, what are these involved in? To some extent. Cap-dependent translation primarily. So to some extent it'd be involved in biosynthesis potentially. And we talked about how when EIF2 alpha is phosphorylated, this usually will block cap-dependent translation and shift translation to a cap-independent mechanism. Okay? PKR's role is to, one of its main roles, when it binds to double-stranded RNA or RNA that is foreign, so it has structures different than our own RNA, it will phosphorylate EIF2 alpha and block translation from occurring. Okay. So again, PKR. So this is just kind of showing that. Regular or unphosphorylated EIF2 alpha allows for translation of cap mRNAs. Phosphorylation of it will block cap-dependent translation or prevent it from happening. Okay. So in this experiment here, they're looking at translation of, I'm trying to remember what virus this is. I can't remember it off the top of my head. But what they were looking at in this experiment was they were trying to look at actual expression of GFP, a cap-dependent mRNA. And they were looking at that at its translation in the absence of VP35 altogether, or two different forms of VP35. One that contains just the full length, or a mutant that lacks what's known as the 3A domain. Okay. So normally what they find, here's low expression of GFP, lots of phosphorylated PKR, which would be the active form. This is measuring total PKR here, okay, just total amounts of PKR in the cell, and then phosphorylated and total EIF2 alpha, and actin as a control. So typically when these blots are looked at, when we're looking at, for example, phosphorylation of EIF2 alpha or PKR, we would compare how much can we detect of the phosphorylated versus the total amount to get a ratio or a fraction. Okay? So if we're looking at this, with no VP35 upon addition of, let's say it's poly-IC here, what do we find? PKR is phosphorylated, EIF2 alpha is phosphorylated. What would be our expectation then? Does translation of capped mRNAs occur? No, probably not. Okay. Now we add VP35 to the scenario. How does this change? Go ahead, Sabrina.
Is the PKR has to be phosphorylated before it gets phosphorylated?
Yeah, and that's something that I didn't point out here. So PKR goes from an unphosphorylated to a phosphorylated form to become active. We'll talk about that phosphorylation step in a minute. So when we add VP35, what do we see happen to the levels of phosphorylated PKR and the levels of phosphorylated EIF2 alpha? Decreased. Expecting what then, Shania? What would we expect in terms of translation? Of capped mRNAs. Does the GFP support that? Yeah, absolutely. So GFP is, we see more capped GFP being translated here than without VP35, right? This 3A mutant, the last one here, is pretty interesting from the perspective of we find a reversion that actually mimics what we see without VP35 altogether. So phosphorylation of PKR, phosphorylation of EIF2 alpha, decrease in GFP expression compared to full-length VP35. What would this suggest? Simple answer here is that, well, without 3A, VP35 loses its function to inhibit PKR activation. Therefore, 3A must somehow be involved in this process. That region of VP35 must be involved. In essence, what they're starting to do here in this experiment is they're starting to break down this protein into smaller parts and determine what are the essential pieces of this protein that are controlling these functions of it. Okay. All right. So how does VP35 do this? It actually has a binding partner, a protein known as PACT. PKR can be activated in one of two different ways. The way that we've talked about most of the time has been through double-stranded RNA. PKR has two double-stranded RNA binding domains and a kinase domain. And when these double-stranded RNA domains are bound by double-stranded RNA, protein opens up. Now we've got that active catalytic domain. First thing that happens, it phosphorylates itself. That's where the phosphorylation step takes place. And now it's active to phosphorylate other proteins. And that phosphorylation there, Sabrina, actually helps to help keep it in this active form. Yeah, helps to keep it open. There's a second way by which it can be activated, and that is with this binding partner PACT. So PACT actually helps to activate PKR in a double-stranded RNA independent mechanism. So one that doesn't require double-stranded RNA. And PACT will bind actually on the active domain, or just outside of the active domain of the protein, changing its shape, and again allowing it to open up. Ebola blocks both of these mechanisms. How? So what's it doing to block the double-stranded RNA activation mechanism? Exactly. Nucleoprotein, or VP35, can help out with this process. The other thing is VP35 will bind to PACT and prevent PACT from binding to PKR. And we would be able to determine this using an experimental method like immunoprecipitation, for example, to show that VP35 does indeed bind to PACT. Now I want to go back to something real quick. And I don't have the evidence right now to say if this is true or not. But when we went back, when looking at the beginning of this, one of the things that I tried to point out here is RIG-I has a partner. Its partner is PACT. I'm wondering, and I don't know if the evidence is there to support this, if the mechanism here is similar to what we see with PKR. If RIG-I can be activated in a double-stranded RNA independent mechanism, by binding to PACT, and VP35 blocks that as well. If that's true, then what it says is that VP35's binding to PACT is actually regulating 2 steps of this interferon response. This interaction here is blocking the activation of interferon beta. And interferon beta, when it is secreted and binds to other cells, one of the proteins it upregulates is PKR. And so not only is VP35 blocking the activation, but it's blocking the subsequent downstream steps that are taking place as well. Okay. So I'll draw this out real quick to try to model what I just said, because this can be a bit confusing. So here's a cell infected with Ebola. So, again, VP35 has a couple of roles. One, it blocks RIG-I signaling. 2, it actually blocks this activation of the interferon beta through its ability to prevent phosphorylation of those transcription factors, IRF3 and IRF7, that help to activate the transcription of interferon beta. And that's a mechanism that we've not talked about, okay, or at least not talked about in detail. VP35 will, so these are all kind of like the upstream steps, steps before interferon beta is expressed. Interferon beta can be secreted. This would represent our paracrine signaling to another close-by cell. That's going to trigger some sort of signal transduction pathway, which would include, ultimately, activation or increased expression of PKR. Well, VP35 blocks that as well. So it's blocking events that would be happening within that cell as interferon beta is being activated, and then steps downstream of that as well. And again, these mechanisms seem to be independent of its ability to bind to double-stranded RNA. I'm going to pause and then see if there are any questions. Shania, go ahead.
Can you explain autocrine versus paracrine?
I can try. I'm not a physiologist, but I'll try. Autocrine means self-signaling. So basically what we would find in that case is not only can this interferon beta activate nearby cells, which is the paracrine, but it can also actually re-stimulate itself if it has an interferon beta receptor on its surface. That's the autocrine. We see this a lot with immune responses where like a cell will become activated and then just kind of continues to like activate itself to keep it going. Sabrina.
So, like normally without Ebola, the point of this pathway would be like that. So like RIG-I to the interferons to the PKR to phosphorylate EIF2 to stop translation.
Yeah. So yes, that is the simple answer. So basically, this is a mechanism. We think back to micro real quick. I know PTSD. you've got immune response. And we talk about innate immunity and we talk about adaptive immunity. And we only focus, when we talk about that, we're only focusing typically on macrophages and NK cells and neutrophils as being part of the innate immune system. Well, there's this whole part of the innate immune system that we don't talk about. And it's what's built into every single cell in our body. And that is these interferon pathways. So we haven't talked about what would actually allow for RIG-I or in great detail about like what would allow for RIG-I to identify this double-stranded RNA. But macrophages, particularly NK and some other white blood cells, even some cells like B cells, will have toll-like receptors or pattern recognition receptors that identify those pathogen-associated molecular patterns. Double-stranded RNA, unbound double-stranded RNA, is a pathogen-associated molecular pattern. RIG-I is an example of a PRR. It is not a cell surface PRR, it is one within the cytoplasm. But it would recognize, since unbound double-stranded RNA is not something that we typically find in eukaryotic cells, that is a trigger for something's not right here. So, for any other RNA virus, this pathway is actually activated. For almost every single RNA virus that we know of, this pathway is activated, whether it be hepatitis C, polio, influenza. In some viruses, like we've seen here, Ebola being one, and even influenza with its NS1 protein, there are mechanisms to kind of dampen down or to prevent this from occurring. And in doing so, it allows for the viral replication to spread. So you may hear from a clinical perspective that some treatment for some infections is interferon therapy. That's basically giving somebody artificially high concentrations of interferon alpha, beta, or gamma to kind of enhance this response through an artificial paracrine or an artificial endocrine signaling as a way to prevent the spread of the viral infection.
So it doesn't have that much to do with translation.
Translation?
It's like the end of
Yeah.
this,
Yes.
but it has more to do with the signaling and
Yes.
and like keep translation.
Yes. Yeah, the goal really is here to like block the translation so that you block the spread of the infection. That's the goal. But viruses have a way around it. Some, they directly affect this pathway. Others use that block in translation to their advantage. Poliovirus encodes an iris. It's cap-dependent translation. It doesn't care if EIF2 alpha is phosphorylated. It's actually a benefit to it because it decreases the competition for ribosomes between itself and the host mRNAs. Tricky little buggers. Other questions? Alright, let me see. Just want to see where we're at real quick. Oh, one thing that I forgot to mention, and this is kind of a side note more than anything else, but there is some evidence to say that VP35's binding to PACT is actually a transition point in the viral life cycle as well. So not only does it benefit the virus in terms of inhibiting the immune response, but it also seems that that PACT will bind to L, which is the polymerase, and that it seems to be a transition from replication to transcription, or vice versa. The evidence for this is kind of not strong right now, so this is an area of active investigation. But it might be, the binding here might be important to the life cycle in terms of the biosynthesis of the virus as well. So before we finish up with the last part, what I want to do is, since we have the time, I want to talk about ERF 3 and 7 real quick. So this outline here represents the cell membrane. Inside of that, we've got the nucleus. ERF 3 and 7 are regulated like most transcription factors, or like many transcription factors, where phosphorylation takes them from an off to an on state in some way. The off to on state here, though, is one in which, I'll try to draw this the best I can. Let's see. And again, phosphorylation is the key here. So let's say that this is ERF 3. When it becomes phosphorylated, it causes a change in the shape of the protein. And a change in the shape of ERF 3 actually exposes what's known as an NLS, or a nuclear localization signal. Anybody want to take a guess as to what the NLS does? It signals for it to go to the nucleus, exactly. It allows it to dock or bind to nuclear pore proteins and allows for ERF3 to translocate from outside of the nucleus to the inside of the nucleus. Once it's made its way into the inside, it now has the ability to bind site-specifically to promoters and it would do so with its binding partner. Usually it's found as a dimer between ERF3 and ERF7. They would recruit the RNA polymerase and would allow for then transcription of interferon beta. So there's a couple of ways in which these proteins can be, well, the mode of inactivation then is just a phosphatase removing that phosphate. Okay. How would we track this localization?
Fish.
What's that?
Can you do fish?
A little bit of fish, but what else could we do to identify the protein, Grace?
Western blot?
Think fish again. Think microscopy.
That's someone else.
IF. Immunofluorescence, right? So immunofluorescence would allow us to identify this particular protein going from cytoplasm to nucleus. So what we would expect to see then is in cases where RIG-I is active, where would we see this, or RIG-I has detected double-stranded RNA, where would we see ERF3? What was that?
Be outside.
It would start outside, and once RIG-I becomes activated, where are we going to find it? It's going to move into the nucleus at that point. Okay. Cases in which this wouldn't occur, mutation to that site that gets phosphorylated. So if there's a mutation of that particular amino acid, like a missense mutation, then we might find that this no longer can localize to different regions. Amelia.
So why would RIG-I go in there again?
RIG-I wouldn't go in there. Sorry, let me rephrase that. RIG-I wouldn't go in there. RIG-I's activation would trigger this signal cascade that would allow for ERF3 to go from this inactive to active form. And as RIG-I is activated, it turns ERF3 into this active form, which allows for ERF3 to make its way into the nucleus. Sabrina.
So with VP35, ERF3 isn't phosphorylated?
Yeah,
So you'd find it in this inactive form?
exactly. So where would you find it? Exactly. That's what we would expect, is if VP35 is functioning properly, we'd expect to see ERF3 primarily in the cytoplasm, not in the nucleus, though. Okay, so today I've thrown a lot of different data. Sorry, Sabrina and Morgan, knowing how much you guys hate this. Mostly Sabrina. Sabrina hates everything. But let's take a step back again. Look at this holistically. What does Ebola virus do to the immune system? So let's just list out, before I write anything down, I want you to take 2 minutes for yourself and just write down what are the ways in which Ebola virus inhibits or regulates the immune response. And then we'll list them out together. All right, mechanisms. How does this virus succeed so well? Go ahead, Grace.
It blocks the recognition of double-stranded RNA.
Okay.
Lots of air.
Okay. And it does so both directly and indirectly. What else?
Expression of
Of what?
interference.
Okay, good.
That's the phosphorylation of IRF.
And that contributes to this previous one.
It blocks a lot of things.
Blocks a lot of things. True. Probably many that we have not uncovered yet either. So this is a pretty good list. And you know, you could hierarchy these, or you could work with this in a couple of different ways. One, you could put them in order. Two, you could put them in terms of hierarchy. Blocking interferon expression is kind of like a top level, and then blocks ERF3 phosphorylation, blocking RIG-I activation would be kind of another one of those sub-levels by which it specifically does that. Okay. One thing that's not listed on here, and I didn't know if we would list it or not, but don't forget that that soluble GP blocks antibody neutralization of the virus. So there are multiple mechanisms, and this is only one of the viral proteins that we know has some, or these are only two of the viral proteins that we know have some effect. There's another one, VP24, also has an effect on the immune system. We just have not talked about that one in relation to the others. Okay? All right. So, everybody, we've talked about plans. The paper for Wednesday is up, so are the questions. Come ready to talk about Ebola virus and mutation frequencies. I think that's what we're talking about.
Do we have to have the final posters submitted?
Final posters should be submitted. I'm asking you guys to submit it to me by Friday night, as well as submitting it to TAC by that point as well.
This Friday?
Yes.
Okay.
Yes. So if you guys get me your drafts tomorrow or Wednesday, I will get them back to you as ASAP so that you guys can have the time to finish up.
In the Blackboard, you put the signed data to 25.